Hello and welcome to this Nature Research custom media webcast titled Tools for Discovery of Novel Targets and Molecules Involved in Neuroplasticity. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by Cellectricon. The rising incidence of neuropsychiatric disorders over recent decades has intensified efforts to find safer and more effective medicines for conditions such as depression. Neuroplasticity plays a crucial role in the onset and progression of symptoms in many psychiatric disorders and provides a handle for new drug targets. This webcast will describe a suite of native in vitro models developed by Cellectricon to study neuroplasticity and quantify structural and functional changes at scale for drug discovery. Through case studies, we will see how the assays can be applied to screen novel drugs and show their mechanisms of action. With us in the studio are two speakers from Cellectricon. We'll begin by hearing from Dr. Paul Karila, who is the head of drug discovery at Cellectricon. And we'll then move on to Susanne Lardell, who is a senior scientist and external collaborations manager at Cellectricon. We'll then open the floor for your questions in the audience. You can ask us a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them at the end of the session today. And now, over to our speakers.
Hello and welcome to our webcast. I'm Paul Karila.
And I'm Susanne Lardell.
Before we get started, Susanne and I would like to thank Sarah for the introduction. Also, we'd like to thank all of our viewers for logging into the event. We hope that you find our presentation interesting and that you pose lots of questions for the Q&A session. As Sarah explained, we will walk you through Cellectricon's latest offering, which is the Services Module for Neuroplasticity Research. In the webcast, you will hear about how a suite of native in vitro models can be useful for discovery of novel targets and molecules involved in neuroplasticity. But before we dive into that, I'd like to give you a short introduction to the topic. So, neuropsychiatric disorders are a major threat to society due to their huge impact on the quality of life for affected individuals and their families. In addition, the incidence of neuropsychiatric disorders has increased dramatically over the past decades. As you can see from the figure, almost 1 billion people were living with mental disorders in 2019. Overall, this is divided fairly equal between females and males, but there are of course variations between the different disorders. The increase is not only due to increased life expectancy. Key threats today include the economic situation, health emergencies, and the growing climate crisis. As an example, two major disorders, depressive and anxiety disorders, increased dramatically as a result of COVID-19. And the pandemic resulted in an increase of 28% for major depressive disorders and 26% for anxiety. This dramatic change occurred in just 1 year between 2020 and 2021. In addition to the suffering on the personal level, researchers estimate that 12 billion workdays are lost every year due to the two major disorders. And the cost for this is approaching $1 trillion. There is therefore a huge need for more research into neuropsychiatry to find safer and efficacious medicines. However, as the graph shows, although 2/3 of years lived with disability in the world comes from neuropsychiatric disorders, only 7% of the funding for global health research is spent on mental health. But I believe this will change dramatically in the near future as judged by our client interactions and the neuroscience-focused conferences we attend. So why is research on neuropsychiatric disorders underfunded? One reason could be that it is hard to find predictable models that are useful in rational drug discovery. For example, the complexity of CNS is difficult to model and the disease relevance of existing models has been questioned. Also, many of the currently applied approaches, such as behavioral studies in animals and electrophysiology, are very labor-intensive and do not have the capacity and reproducibility required to identify novel mechanisms and drugs. But from our perspective, as experts in high-capacity screening in advanced in vitro models in other disease areas, we saw an opportunity to do exactly that. namely to establish models that could be valuable for our clients working on neuroplasticity projects in biotech and pharma. By combining drug discovery expertise with fit-for-purpose technologies, we are creating highly sophisticated in vitro assays and disease models. For example, we support our clients in their projects by validation of novel targets involved in protein aggregation using high-content imaging assays. We also perform large-scale target and lead discovery screening in, for example, Parkinson's, ALS, and chronic pain projects. The methods we use can measure effects of compounds and target manipulations on both morphological features using high-content imaging and functional readouts, as well as gene and protein expression. The functional readouts we typically look at are cytokine release, peptide release, neuronal excitability, and synaptic function. For neuronal excitability and synaptic function, we use our proprietary optical electrophysiology platform, which I will talk a bit more about later. When it comes to disease modeling, we can either add chemical insults in vitro, or use genetically modified animal tissue, or use patient-derived human iPSCs. Also worth mentioning is that we are working on neuroinflammation, where we are establishing complex neuron microglia astrocyte co-cultures for modeling of neuroinflammatory conditions. We're now using the experience we've gained for example, target identification and target validation projects in Parkinson's and chronic pain in our neuropsychiatry efforts. Many of the currently used medicines and psychedelic drugs were discovered by chance, and it has therefore been difficult to perform rational drug discovery. The good news is that many of the currently used medicines are also active in our in vitro models, so there is a relatively good back translation. This is hopeful, since the reverse might also be true, that is, molecules discovered in these models may also work in man. This makes it reasonable to believe that a phenotypic screening approach could also add value here, for example, by screening extracts from plants used in traditional medicine. But more importantly, since there is a clear connection between neuroplasticity and existing as well as emerging drugs, there is also an opportunity for target-based drug discovery. I will use BDNF and its receptor TrkB as an example. Recent studies indicate that both conventional antidepressants, fast-acting drug ketamine, as well as psychedelics act, at least in part, through activation of TrkB receptors. BDNF is also active in our in vitro assays, and we can see effects both on the structural and the functional level. One could therefore use native neuronal assays to screen libraries of TrkB targeting molecules against readouts relevant for neuroplasticity. Other tool molecules and drugs that are effective in neuropsychiatric indications, such as ketamine, are also active in our assays, and it is possible to investigate if BDNF is part of a mechanism of action by, for example, performing receptor blocking experiments or knockdown experiments for these molecules. Together with our ability to generate high quality and high capacity in vitro models, this provides a handle for drug discovery to identify novel targets and molecules that affect neuroplasticity. Due to this opportunity and the growing interest from our client base, we have during the last couple of years established and validated in vitro models for neuroplasticity with leading clients in the field. We've now packaged these into a new service module that we launched during the fall, and we're now making the module available to other interested clients. The assays in the module are currently based on rodent CNS cultures, but we're also working on humanized versions of these. The assays cover important neurobiological events related to neuropsychiatric diseases, all the way from neuritogenesis, that is the process by which neurons form neurites, to synaptogenesis, that is the formation of connections between the neurons, and how this results in changes in neuronal function. Susanne will talk about the neuritogenesis and synaptogenesis assays in more detail in a while, but I can tell that they are all endpoint assays based on high-content imaging and analysis. To investigate transport of proteins and organelles along neuronal processes, we are using our proprietary microfluidic platform, which is a high-capacity version of a Campanelle chamber where we can evaluate 100-fold more drugs or experimental conditions compared to similar devices. And we can investigate how drugs, for example, are affecting transport of synaptic proteins. Before handing over to Susanne, I'll spend a couple of minutes on how we can measure neuronal excitability and synaptic function. We believe functional assays are important since all structural changes of relevance will eventually manifest as changes in neuronal excitability and synaptic function in disease states. We therefore spend a lot of effort on these readouts, establishing assays on our proprietary optical electrophysiology platform. The uniqueness of a platform comes from the fact that it can produce a homogeneous electric field across the cell cultures in each well of a 96 or a 384 well plate. This combined with imaging of the response of the electrical excitation using optical probes such as calcium or voltage indicators. We can therefore measure, for example, drug-induced changes in excitability in a high-capacity fashion. We have vast experience doing that both in phenotypic and target-based client projects using both rodent and human backgrounds and with both peripheral and central neurons. All of the assays in the module can be used standalone, but we believe that a broader data package that is collecting data from more than one of the models will aid in translation both to the in vivo situation and to human testing. As discussed, two key changes that occur in neurological diseases are alterations in neuronal excitability and synaptic signaling. In the synaptic function assay, we apply high-density rodent cortical cultures in the 384 well format. With time, the neurons in the culture will grow longer processes and become synaptically coupled. We use our optical electrophysiology platform to measure effects on synaptic function. In this setting of the instrument, we combine electric field simulation with imaging of the response to the electric field using a calcium indicator. The electric stimulation results in a brief calcium transient, and the example to the right is a raw data trace with 2 consecutive stimulations. Most commonly, drugs will affect the amplitude of the calcium transient resulting from the electric field stimulation, but also other features such as peak width will be affected. The assay is validated with molecules having a wide range of mechanisms of action, making it suitable both for target-based and phenotypic screening approaches. As mentioned, the platform was designed for high-capacity screening, which in this case means that we can support screening for up to 100,000 compounds in single concentration. To the left is an example of a QC log from a recent screen where we tested 50 small molecules in concentration response format. Almost 90% of the plates passed QC, that is, the assay window was larger than 0.3, as marked by the dotted line for the Z prime. This shows the quality and reproducibility of the dissections and cell culturing that the team at Cellectricon achieves. And finally, we can also obtain early mechanistic insight in the synaptic function assay. Here, ketamine behaves like an NMDA antagonist, as we can see from the reduction in the response. When looking carefully at the electric field stimulation response, which is the red trace in the right figure, you can see that there is a residual signal that is not blocked. This is due to that the signal is derived from both neurons that are directly excited by the electric field and from second-order neurons that are connected to the electrically excited neurons. On the other hand, ketamine fully blocks the spontaneous activity, which is another feature that we can measure as seen from the black curve. This indicates that the drug acts on synaptic communication as opposed to excitability. If we look at another drug, for example, a sodium channel blocker like lidocaine would have identical curves for the spontaneous activity and the electric stimulation evoked response, just like the black trace. So, to summarize what I've shared about the synaptic function assay today, it's a key assay in the module since it can provide information on a functional parameter that is affected in neuropsychiatric disorders. It can detect the activity of compounds with varying pharmacology. It has a high capacity, and together with above, this means that it is suitable to use for identification of new classes of compounds, using both target-based and phenotypic screening approaches. And the assay can also be used for elucidating mechanism of action. We believe that the assay provides value by bridging the gap between simplistic assays in overexpressing cell lines and in vitro models. I will now hand over to Susanne, who will tell us more about the high-content imaging assays in the neuroplasticity module.
Thank you, Paul. I'm pleased to be talking here today, and thank you all for joining us. Today, I will focus on 2 assays in the neuroplasticity module, both based on high-content image readout, the neuritogenesis assay and the synaptogenesis assay. But before going into details about the assays, I would like to give you a brief overview of the events evaluated in these assays and the importance of understanding more about these processes. So neuritogenesis, which is the process by which neurons sprout neurites, and synaptogenesis, which is the formation of synapses between neurons in the nervous system, they both play a crucial role during brain development, but also throughout life, and are important for brain plasticity, which is the brain's ability to adapt to changes. Abnormalities in neuritogenesis and synaptogenesis have been linked to neurodevelopmental disorders, such as autism and intellectual disability, neuropsychiatric diseases, such as depression, and neurodegenerative diseases, such as Alzheimer's and Parkinson. And studying these processes are therefore vital for understanding brain function, brain development, and importantly, to provide therapeutic insights for the treatment of neurological and psychiatric disorders. With that saying, there is an urgent need for in vitro models that can be used to study these events and that can be applied in a drug discovery context. I'd like to start off telling you more about our neuritogenesis assay. So this is a medium throughput assay, and it's based on 384-well plate format. And here we use primary rodent cortical cultures that are sparsely seeded in order to enable single-cell analysis of neuritogenesis. And in these assays, compounds' ability to affect neuritogenesis is evaluated in a concentration response format. And after 9 days in vitro, the cultures are stained with different markers to enable visualization of nuclei, neuronal soma, and neurites. Structural effects induced by compounds on neurite branching and length is then quantified using high-content imaging and analysis both on the populational and single-cell level. The assay has the ability to differentiate compounds both based on efficacy and potency, and it's therefore possible to rank order compounds, enabling prioritization for inclusion in downstream models. And now to some compound effect data from the assay. So we have evaluated an extensive set of tool compounds in this assay, and we found that we consistently could quantify compounds modulating effect on neurite branching and length. Importantly, we found that compounds with broad range of mechanisms were demonstrated to affect neuritogenesis. And with these images, we want to illustrate that the effects on neuritogenesis can clearly be visualized. And to the left, we have an image of rat cortical cultures treated with the vehicle control, and the cultures are stained with beta-3 tubulin. The middle image represents a culture treated with DOI, which is a 5-HT2 receptor agonist, and it's a positive control in the assay. And you can clearly see the effect on neurite outgrowth. In this right image, we have treated the cultures with donepezil, which is an acetylcholinesterase inhibitor, and it's used to treat symptoms of early Alzheimer's. And here the effect is even more pronounced. So in these examples, the compound's effect on neuritogenesis is very obvious. However, even where the effect is not detected by eye, modulation on neurite parameters can easily be quantified. And now to some more compound effect data from the assay. So these graphs represent typical concentration response data generated from the assay, and all data is normalized to vehicle control. And if we start with the graph to the right, we see concentration-dependent effects on total neurite length plotted for donepezil, and we can see a clear increase with increase in concentration. In the middle image, we have treated the cultures with fluoxetine. It's a selective serotonin reuptake inhibitor, and the data here is the quantified data for the same parameter, also increasing neurite length in a dose-dependent manner. Moreover, we can study mechanism of actions of compounds, and it's illustrated in this image to the left, where we have evaluated the two enantiomers of ketamine, and we could clearly identify the S enantiomer being far more potent in the assay. And now to some screening performance from the assay. So as Paul mentioned earlier, the quantification of drug effects on structural neuroplasticity has been challenging due to assay variability and also low throughput. And here we would like to demonstrate how we have addressed this with excellent performance in our assay. So if you look at the bar chart to the left, you can see screening data for the positive control in the assay DUI, and the data represents the assay window in percent effect normalized to control. And here we have graph data from 60 plates with cultures prepared from 15 individual preparations. And out of the 60 plates, 4 plates failed QC. And so the data demonstrates consistent assay performance over time, and for the last 50 independent test occasions, over 90% passed the quality control. In addition, the assay shows excellent reproducibility for compounds that have been tested more than once in the assay. And in the graph to the right, this is demonstrated with a correlation plot for around 30 compounds that were tested twice in the assay. And as you see, we have very good correlation between the two test occasions. And to conclude, the neuritogenesis assay is both reproducible and sensitive with excellent assay performance, which makes it fit for purpose for long-term screening programs. To summarize what we have showed you today regarding the neuritogenesis assay. So the assay is sensitive and highly reproducible. It can be used to differentiate compounds based on both efficacy and potency, making it possible to rank order compounds for inclusion in downstream models. And as demonstrated, the assay may be used for the discovery and characterization of compounds modulating neuritogenesis with a broad range mechanism of action. The assay is now being employed in continuous screening in a lead optimization program of compounds derived from psychedelics. What we have not gone into detail about today, but I also want to highlight, is that the assay can be used for complex pharmacological investigations. For example, for evaluation of combinatory effects of compounds, as well as to determine mechanism of action. And from here, we will continue on to our synaptogenesis assay. So this is an overview of the assay. And in short, the assay is a complex imaging assay for quantification of changes in pre- and post-synaptic proteins. And it's based on co-localization of the markers synapsin-1 and PSD95. And for this assay, native rodent cortical cultures are prepared with high reproducibility in a 384-well plate format. And the cultures are prepared at a high density to allow the neurons to become synaptically connected, and this then to enable studying synapse formation and loss. Compounds are evaluated in concentration response format, and several readout time points can be evaluated, but 7 to 14 days in vitro are the most common. Following fixation, the cultures are stained with pre- and post-synaptic markers, as well as markers for nuclei, soma, and dendrites of neurons. This is followed by high-content imaging and analysis of individual markers, as well as co-localization of the markers. And the monoparametric readouts that we obtain can then be used for concentration response determination. And this is exemplified here with BDNF in this graph versus vehicle control. In addition, more advanced multiparametric analysis can be performed, and this in order to evaluate phenotypical features of compounds. So during the assay development, a range of antibodies targeting pre- and post-synaptic markers were evaluated for their immunocytochemistry, compatibility, performance, and specificity. And based on the outcome, we selected the markers synapsin-1, which is a presynaptic marker, PS95, which is a postsynaptic marker, and they were further evaluated using high-resolution microscopy to confirm the co-localization of the markers as well as localization along the MAP2 dendritic structures. And this image is captured with Cell Discoverer 7, and it represents a maximum projection of 7 planes. And you have MAP2 in red, the presynaptic marker synapsin 1 in purple, and the postsynaptic marker PS95 in green. And if we zoom in a bit, we can more clearly visualize the co-localization of the pre- and postsynaptic markers. Here encircled, indicating that synapses are formed. And this is then further illustrated in this 3D video clip. Then for quantification of synaptogenesis on a more regular basis, imaging is performed using ImageExpress followed by analysis in MetaExpress. And with the analysis set up, we can, for example, identify number of viable cells, we can quantify the MAP2 network, we can quantify number of PSD95 and synapsin 1 puncta, and the co-localization of the markers within the MAP2 network. However, for the markers analyzed, the total number of output parameters exceeds 400, which provides an excellent opportunity for multiparametric analysis. And I will talk a bit more about that later in this presentation. And in this image, which is captured with ImageExpress in confocal mode and water immersion, PSD95 is displayed in green and synapsin 1 in orange. And after a range of analysis steps, which I will not go through today to save some time, we can identify and quantify co-localized pre- and postsynaptic markers along the MAP2 network. And in this image, it's displayed in pink. And for this assay, we selected BDNF as a technical control due to its neurotrophic properties and literature precedents for increasing synapse density in vitro cultures. And if we, in these images, compare the BDNF treated cultures to the right here with vehicle control to the left, we can see a clear increase in number of synapsin 1 as well as PS95 puncta. And this is consistently observed and quantified for BDNF within the assay. And after treatment with some compounds, for example, ketamine, fluoxetine, and psilocin, clear phenotypical differences were observed for the localization of the synaptic markers. And these are distinctly different from that of, for example, BDNF. And in this example with ketamine, we can see a clear increase of synapsin 1, but also a redistribution of the marker in comparison to the vehicle control. And as I mentioned previously, the total number of output parameters with regards to number, sizes, intensities, and shapes exceeds 400. And this provides then a perfect opportunity to perform a multiparametric analysis. So to further evaluate the phenotypical differences that we observe between these compounds, we extracted all the parameters and performed a principal component analysis for select compounds. And the output principal components were then used for clustering, potency determination, etc. And we found that using this multiparametric analysis, we could identify both phenotypical differences, but also similarities between compounds. And this is illustrated in this left image. So at the top, we have two compounds that are clustering, indicating phenotypical similarities, whereas the compound at the bottom, and it's 4 different concentrations of this compound, is distinctly different from those at the top. And compounds were also shown to cluster differently in the PCA space. And that is illustrated in this middle image, where we have the vehicle control in red. We have a ketamine here in purple, and we have psilocin in green. And in addition, the potency determination was possible. And this is illustrated with these graphs to the right here, generating very neat data. So here we have ketamine and psilocin. And to sum up what you have learned today from the synaptogenesis assay. So the process from tissue generation to analysis of synaptogenesis and the assay concept has been validated, and we are confident that we are staining and acquiring images of synaptic structures. And our technical control BDNF further validates the assay concept by consistently increasing co-localized pre- and post-synaptic structures along the dendrites. And although the effects on synaptogenesis can be quantified for many compounds using the monoparametric readouts, the proof of concept that we performed using the multiparametric data analysis indicates that this can be used for more advanced phenotypical evaluation of compounds, as well as for potency determinations. And we have now walked you through 3 of the assays included in the neuroplasticity module. And the assays can, of course, be used standalone, but we think it's most efficient to profile compounds across many assays. And I will now provide an example of that. So in this table, we have summarized the effects for compounds tested in all 3 models. The tested compounds are a mixture of psychedelics as well as clinical compounds that are used for treatment, for example, depression and Alzheimer's. And as you can see here, different compounds are active in different models. So whereas BDNF and ketamine are active in all 3 assays, effects of other compounds, such as psilocin, can only be detected in the synaptogenesis assay. And this may then point to a differential mechanism of action, indicating that the assays in combination could be a valuable tool to identify and elucidate the functional molecules with novel mechanism of action.
Thank you, Susanne. Before the Q&A session, I'd like to sum up the key points from this presentation. Neuropsychiatric disorders are a major challenge, but we see a way forward based on the emerging mechanistic knowledge and our in vitro drug discovery and neurobiology experience. The development of our assays was driven by the lack of validated in vitro models for studying neuroplasticity. We therefore established and validated a set of assays together with leading clients in neuroplasticity research and packaged these assays into a new services module that we now reach out to new clients with. The module has several applications and can be used in target and lead discovery projects for identification and prioritization of new candidate molecules, for supporting the structure-activity relationship, and for elucidating mechanism of action, as well as for target discovery. The included assays take both anatomical and functional aspects and of neuroplasticity into account, and the assays can be used standalone or in combination for a broader profiling, and to facilitate translation to in vivo and human testing in drug development. Lastly, we would like to thank you all for listening. Please reach out to us if you want to find out more. We're now looking forward to your questions.
Thank you very much to both Paul and Susanne for that presentation. It is now time for our question and answer session. Please, if you have a question, type it in using the Ask a Question box and press submit, and we will attempt to answer all your questions, or as many of them as we can today. So moving on to our questions already, I would like to begin with this question, which is for you, Susanne, and it centers on the table you brought up at the end. All the psychedelics in your table seem to be active in the synaptogenesis assay, except for the 5-MeO-DMT. Do you have any theory around that?
Yes, Yeah, so psychedelics, of course, act through many different mechanisms to induce neuroplasticity and also have different affinities for the receptors that are involved. And many psychedelics act and promote synaptogenesis in a 5-HT2A dependent manner. But 5-methoxy-DMT is unusual in that sense among psychedelics, since it has much higher affinity for the 5-HT1A receptor. And this receptor is mainly inhibitory and then tend to have the opposite effects compared to 5-HT2A. So that might be one explanation.
Okay, thank you very much for that. Our next question is for you, Paul, and it asks about your data on rodent models. All the data that you've shown, I think, are based on rodent models. If so, from a clinical point of view, how relevant are those models?
Thank you. Yes, you are correct. All the data that rodent models. But having said that, many of the test compounds, as you may recall from the table that Susanne showed, are actually active. And that is, as an example, then we have molecules as psilocin, ecstasy, ketamine, other NMDA receptor modulators that are actually on the table for psychiatric indications are active in the model. And this, of course, gives us hope, since it could also be true that there could be a reverse relationship, of course, that models, compounds that are active in our models are also working in man eventually. And of course, we are testing a lot of client molecules having different mechanism actions. And it will, of course, take a long time until we know the definitive answer to that question. Would they work in man? But of course, our hope is that they would, right?
Yep.
Yeah, first the mouse model and then the man model. Okay, so our next question, Susanne asks about psychedelics in vivo. These are mostly fast-acting. How do you address that, or is that something you can address in your in vitro assays?
So my short answer would be yes. If we look into the different assays, so for the synaptic function assay, here we mainly look at fast-acting mechanisms. We usually incubate the compounds for 1 hour with the cultures. And here we have effect, for example, ketamine, which is a fast-acting psychedelic. For the neuritogenesis and the synaptogenesis assay, these assays were developed to detect compounds with broad time course for inducing neuroplasticity. And here we typically incubate compounds up to 6 days. But also in these assays, we can detect the effects of fast-acting compounds.
Okay, thank you very much. A reminder to our live audience, you do still have time to ask a question. To do so, please type your question in where it says type your questions here, and then press submit. Paul, have you ever used human stem cell-derived neurons in any of these assays?
No, we haven't done that yet, since it's a relatively new offering for us, but we do have long experience using iPS-derived neuronal cultures for other indications, such as chronic pain and Parkinson's work, as an example. But having said that, of course, if a client would be interested in investing in doing that with us, we would be all ears and be interested in doing that.
Fantastic. Thank you so much. Susanne, ketamine and memantine are both NMDR receptor antagonists. Do you have any thoughts on why they act differently in the synaptogenesis assay?
Yes, so ketamine, which is a fast-acting antidepressant, acts by inhibiting NMDA receptors, but it also acts by triggering downstream signaling pathways, such as triggering BDNF protein synthesis. And memantine, on the other hand, has limited efficacy in inhibiting these receptors, and therefore does not trigger the same pathways. So I think that could be one explanation.
Okay, thank you very much. Our next question I'm going to direct to you, Paul, and it asks if you have any other assay instead of microscope-based assays.
So a part of my presentation was on our functional assay, which we use in what we call our optical electrophysiology platform. So that's, of course, one that comes into mind here, which is a high capacity electrophysiology type of assay. We also have MEA, multi-electrode array assays, where we can study synaptic connectivity between neurons in a network in a more detailed manner, as an example.
Okay, what about cytokine release?
Yes, that's another thing that we actually are doing. Again, limited experience in this disease area or within this module, but that's something we have experience from, both from our pain and neurodegeneration experience.
Fantastic. Okay, thank you very much. Our next comment, great presentation. There have been some interesting clinical developments when it comes to evaluating treatments for neuropsychiatric disorders. Now, how would your models have been valuable for the identification and development of drugs such as those? And perhaps you might know more clearly to which drugs they might be referring in the question.
Should I take that one? Yeah. So if we start with clinically approved drugs, Spravato from Janssen, we have Sage's and Biogen's positive allosteric modulator for GABA A. And then in phase 3, I believe we have MAPS that has now changed to Lykos. They have an MDMA analog. And then we have Compass's psilocin analog. Those are all examples of mechanisms, drugs against mechanisms that are actually active in our models. So I do think that there is a good relevance. Overall, there are more than 150 clinical trials right now for psychiatric indications. So of course, I don't have an overview on all of those, but there are some interesting mechanisms that I think are interesting. For example, TRP channel antagonist, TRP A or M4, M5, for example. And then we had a TAR1 agonist. Would we have detected these in our assays? We don't know. But of course, we could envision helping clients to find novel mechanisms, novel drugs like this, since we do have large experience using tools such as shRNA knockdown of targets in our in vitro models, for example, in chronic pain and Parkinson's disease. So that's something that would be, of course, interesting. Hope that answered your question.
Thank you very much. Our next question is for you, Susanne. Are there any advantages of using rat cortical cultures rather than hippocampal cultures?
Well, for us, it was a rather straightforward approach to start off with cortical cultures, since this is a well-established system that we had experience for, I think, 10 years. And also, when we started developing these models, it was important to use brain regions where we know we have effects of psychedelic-like structure compounds, such as cortex, and also that highly express 5-HT2A receptors. But of course, if there's an interest in evaluating these assays using hippocampus cultures, that is something we could do in a client-funded project, I would say.
Okay, thank you very much. A reminder to our live audience, you do still have time to ask a question. To do so, please type your question in where it says type your questions here, and then press submit. Okay, So our next question asks how these assays will help to define the mechanism of action of different CNS targets in a variety of diseases. Now, either of you might like to answer this question. Susanne, would you like to take a first stab?
I think that maybe you might have a better answer to that.
Well, I was thinking about the antagonist-agonist experiments that we're doing right now, maybe something around those.
Yeah, so what we can do, for example, in the neuritogenesis assay is to evaluate if the effects we see on neurite branching and length for compounds, if we can inhibit those by doing combinatory experiments with blockers for specific mechanisms. So that is definitely one thing we can do.
And perhaps along those lines, combine, I mean, typically, at least psychedelics are typically agonists, so combining them with lentiviral SH knockdown in a systematic manner of different targets, maybe.
Interesting. Okay, thank you very much for that. Our next question asks about neurite retraction. Could that be beneficial for mental health? What would your view be on that, Paul?
I mean, typically, I would say that in mental diseases, there are too few functional synapses, in my understanding at least. So I'm not well enough read up on whether there are diseases where that could be the case. But we could, in that case, we can study those types of events for sure. But I'm not knowledgeable enough to understand the relevance there.
Okay, so our next question is for you, Susanne. Can you perform shoal analysis in your neuritogenesis assay?
Yeah, so a shoal analysis is used mainly to measure arbor complexity, and that is definitely something we could do with the analysis workflow that we have set up. But besides that, we can analyze a lot of other aspects on neuritogenesis. And I would say that compared to a shoal analysis, our workflow is much less biased and with reduced variability compared to that.
Okay, thank you, Susanne. Our next question asks about the rodent. Are you using adult rat neurons or newborn rats? Could you clarify that for us?
Suzanne. Yeah. So we're using embryonic neurons.
Okay, wonderful. Thank you very much for that clarification. Okay, well, on that note, I think we are going to close for today. I would like to thank Dr. Paul Karila and Susanne Lardell very much for being with us and for answering our audience questions today. I would like to thank webcast sponsor Cellectricon and of course you, the audience, for tuning into this webcast today. You will be redirected to a short survey. Please do take a moment to complete the feedback questions. Remember, you can watch this webcast again at any time at nature.com/NatPort Webcasts. That's NatPort Webcasts. Thanks for watching, and I hope you'll join us again soon.
Thank you. Bye.
Thank you.